So I've gathered a few questions from the chat, and it's not all questions. I'm sorry in advance if I didn't get to anyone's question, but let's just review these ones fast, answer them, and then start right away. So this first question here is regarding ClinVar data and ClinVar submissions, and do we trust other people's submissions? Specifically, this is, what do you think regarding the usage of internal data based on ClinVar submissions? Meaning, for example, if Invitae claims variant is likely pathogenic because they had a de novo status, can we use it to apply to our internal classification? Well, the answer is a bit complicated here, but the thumb rule is that you need to trust the lab that is submitting this variant, is classifying the variant. I go by trust the large labs that are submitting and classifying variants frequently, like Invitae and GeneDx and other large labs. But you always have to be skeptical from the other side. So we write them many times. We email these companies. You're entitled to do that based on the submission. If you don't have enough information, you can just email Invitae directly or any other submitter and ask for additional details for you to use their information in their submission. This is sometimes required. The next question was, is there currently a way to access the non-cancer database or dataset in NOMAD4? And the answer for that is no. As far as I know, NOMAD4 does not contain specific data sets that you can choose among. In the previous data sets or previous versions of NOMAD, you could choose control and non-cancer, non-neurological. But then now it's not possible, as it's all one large data set. The next question regarding NOMAD was, how can we improve NOMAD in the Middle East? Well, the basic answer is that this is a hard process to go through, but NOMAD is aggregating data, sequencing data from several projects worldwide. Obviously, you can reach out and they would probably be glad to have more data if you have it available. The problem is funding and projects to fund the Middle East population sequencing data. And eventually this would happen. We see also lacking information from Africa and other countries. So this is not only specific for it's not specific for the Middle East. The next one was, would we use Missense Z-score frequently? I only use PLOF. Now the PLOF measurements only applies on a loss of function variant. But when you're trying to assess the Missense variant, you have to use the Z-score. The Z-score reflects the number of Missense variants that are expected, that are observed versus expected in a given gene. It does hint on whether the missense is the mechanism of disorder for a specific gene. And usually missense would cause gain of function, although we do know that some or certain missense variants could also lead to a loss of function. But basically, this would all fit in the ACMG classification, and you would get specific classes on the Z-score, NP-LOF, and how to use it for ACMG classification later on. The next question was, does ClinGen provide dosage based on the variant? Well, the answer for that is no. The dosage sensitivity parameters that are provided by ClinGen are based on the gene level and not on the variant level. And usually, different variants in a single gene would have the same dosage sensitivity. So this actually makes sense. It's on the gene level, not on the variant level. The last question is, on an average, how much time it takes for you to check all parameters while closing a case, supposing an old exome sequencing? The answer for that really depends. If we have an easy case, I would say, like there is a clear diagnosis, this could take just a few minutes. All the parameters that I've showed you are found in the platform. So it's not like you have to wander around and go Google and go through different websites, because it's all concentrated on the same platform. So this is also very important to mention. It could take some time to analyze a single case, but not because that we have to check all parameters, just because there isn't a clear diagnosis for the patient. And for such cases, we have to classify many different variants of unknown significance and to try to clinically correlate them with the patient's phenotype before we can close the case. In such cases, and one analysis could get to an hour, 2 hours, and sometimes it lasts 2 days when you review the case again and again. So I hope I answer your questions, the main ones. And now I can jump in right straight to the cases from the clinic. So the first case that I want to present to you starts with a story about a child named Daniel. Daniel is 2 years old and he's a male child. And the family history of this child, he is the first and only child of 2 parents that are non-consanguineous. And there is no significant medical history for the parents. The child itself, Daniel, in terms of perinatal history, there is nothing very important to mention. This was an uncomplicated pregnancy and delivery. During neonatal or childhood, there were some findings. First, it was microcephaly, which is a small head, facial dysmorphism, which we know that characterize many, many different syndromes. Also, there was corrected atrial septal defect, which is I think the most common heart defect that one can find. And there were delayed milestones also. In terms of growth, Daniel presented with failure to thrive. We usually use the abbreviation way of saying FTT. So FTT is failure to thrive. He also had hyperreflexia and radioulnar synostosis. So this is overall the lists of phenotypes that were provided by the physician of Daniel, by the referring physician. And the physician referred Daniel for an evaluation for a genetic syndrome with microcephaly and cardiac abnormalities and developmental delay, obviously, because there were some delayed milestones in facial dysmorphism. And the overall clinical assessment of short stature, low weight, and small testis was also mentioned in the referral letter. So overall, this is a classic case of a child with a specific phenotypes that arrives or comes to a genetic counseling session or just to see a geneticist. And the geneticist would determine or would refer the family and the child for a genetic test. In such a case, this could start with a CMA, which is the chromosomal microarray, or CGH array, depends how you want to call it. And then if this would be negative, then the next genetic test, or even in parallel, would be an whole exome sequencing or genome sequencing, really depends where you are. In this specific case, the child was referred for an exome sequencing, and it was a single exome sequencing and not a trio one. So with that, for the first time, I want to open the system, just show you live how do we analyze these cases. So before I jump in to the actual case, I do want to take a few minutes to describe Genix on general, because for those of you who are interested to have a hands-on experience, you have an account, your own account, and you would need to download the files, the VCF files, and upload them to Genix, run them through annotation, and also write the phenotypes and run the analysis yourselves. So here is my chance to train you. So this is the main dashboard of Genix analysis. On the left side, you have a navigation tool, which is very helpful. You can jump to all the analysis in your account, all the VCF samples that you've uploaded, all the reports if you issued any, all the subjects, and we also have a boost monitor here to monitor a variant of unknown significance through time. There is also, in terms of features of the platform, there is a variant browser in which you can, after you're aggregating enough data, You can query your database for specific genes, specific variants, specific subjects and phenotypes. So all the data that you're gathering or aggregating in your own account is queryable by the variant browser. There is also the settings and other features, but the settings is one of the most important area in Genix, because from this tool here, from this screen here, you can change and monitor many of the features of the platform. You can customize the account to suit your own needs and the needs of the lab. Specifically, you can create some protocols here. So this is just default lists of protocols. If we just review them for a second, we can see that there are exomes and genomes. There are long reads protocols, somatic analysis protocols, pharmacogenomics, couple screening if you want to compare a couple and to search for shared common carrier status for this couple. There is also a mitochondria analysis and CNV analysis. And basically every account looks different. You can create and open as many protocols as you wish to fit or suit a specific sets of analysis and tests that you're offering in your own lab. Now for the course, specifically, we're going to use the standard protocol, exomes, whole genome sequencing analysis. If there is a copy number variations, this would be included inside. So far, we only learned about SNVs, single nucleotide variants, and this is why the next few cases are going to focus on SNVs. During the course, Ali is going to provide some classes on copy number variations and structural variations, so you would also get the chance of capturing these types of variants and analyzing them. So if going back to the main dashboard, in the main dashboard on the upper panel, you have the lists of open analysis. An analysis could be assigned to a specific user in the account. And there are statuses, so you can open the analysis, you can put it in progress or close the case if you want to know what is the current status of a specific analysis. On the left, on the lower panel here, there are the lists of the recent VCF samples, and you can jump between pages. Now, this specific account is dedicated for the course, so we don't have a lot of allele frequencies here or database that was already aggregated, only the samples that are dedicated to the course. All the samples that we're going to provide you and that I'm going to show are synthetic samples. These are not real individuals, as we understand the problems that we have with sharing the data of real individuals. For that reason, because we want you to have a chance to train yourself and to have an hands-on experience, we created these fake VCF files that allows you to almost have the real-time experience of analyzing a patient. Although the VCF is fake and the individual is fake, all the variance comes from a random mixture of many different individuals or variants. So specifically, we started with a case number 1, and this is actually the first case here that we see here. The sample ID is 29, and I've already opened it. So if I want to open, I just click here on Analyze, and then this would send me to the variant dashboard. If you want to start your own case, you want to upload the VCF sample for the first time, and this is what you would do. This is the homework that we are going to give you today. Then you have to go to subjects. You can create here a new subject. So just create it very easily. You can name it as you wish. There is no specific name. It doesn't really matter. You can provide the gender if you know the gender, and we are going to provide you referral letters, full referral letters with gender and phenotypic information for all the samples. So you can just review this and apply any information that you get from the referral letter. Once you open the specific subject, you can upload a VCF. So if you click on it, this is the subject page, and you can click on the VCF samples here, add, and just upload the VCF files that we will give you a link to their download. So you upload the VCF files and then just fill in a lot of information that is important in terms of, for example, the genomic build, etc. I don't have an example on how to customize or provide details on the VCF level here, but we are going to share with you a detailed document on the specific parameters that you need to fill in. Once the VCF is uploaded, for example, this one, you would send it through annotation, annotate here, and then this would be ready to analyze. All these instructions would come in writing, so don't worry. This is going to be super clear and easy. Once you have the analysis, you can just click on it and open it. This would bring you to the analysis page, for example, this one. And then in this analysis page, there is a lot of information on the version set that we used. If you click on it, you would see which versions we used for each and every annotation source, and I've showed it to you in the previous class. You would have the genetic models that are combining or that are found inside this analysis and other type of information. Now, going back to our case now, let me just remind you, we are talking about a patient named Daniel. He's 2 years old and he has a medical history of overall macrocephaly, facial dysmorphism, and other features. Now, when you open an analysis, you have to, or you want to provide the phenotype information. And let's see how we do that. So I've clicked Analyze on the specific case, and this is the variant dashboard. Very quickly, every row here provides me detailed information on every variant that comes from the VCF. As you remember, we discussed annotation sources, and we see here different sections. The first one is genomic and genetic data. Then we have an ACMG classification system. After that, we have the variant calling Q&R, clinical evidence, in-house panel to recapitulate all the information that you have in your own account, effect and prediction, and then frequency. Because there is a lot of many annotation sources, too much to show all of them in one screen, you can open this arrow here and get more information per variant under each and every section. So for example, under effect and prediction, if you expand this arrow, you would get information from CAD score, impact, PLI, Reveal, SpliceAI, and all the other annotation or prediction tools that we are using. And I've elaborated more in the previous class. So every row is a variant, and that is very clear about different columns providing different annotation sources. On the left side here, there is a panel that allows me to report variants. If I want to report a variant, I would click here and I can fill all the information, and this would send this variant into the report. So of course, the reports are automated. You don't need to copy paste anything. The reports are just being created while analyzing. On the orange bar here, which calls filters and tools, if I click on it, I would get a filtering capability for the platform and also the lists of phenotypes, the disease frequency that I'm predicting. And for severe disorders, I would use 0.1. For common disorders, I would use just 1%. There are also panels here or options to add a gene panel, a gene list, or a variant map. Now, these are features that are dedicated for virtual panels. Usually, most labs would sequence the exome sequencing, but then would only report a set or a panel of genes. And this is something that you can easily create. Here in this account, there are only 2 panels, but you can assign them and then analyze them. And from the settings, you can easily apply more and more or generate more and more panels. And we also have the panel app website that helps you to create these panels. So this was really quickly just to review the platform with you. we also have many demonstrations live on our website. So if you want to know more, if you want to know how to upload a sample and you want to have the full tutorial, just go online to our website and it's all there under the video library. Filters. So here there are all the filters. If I click on the plus, I would see which filters are applied right now and which filters are not applied. I can actually also deselect all and save it, and I can just cancel. From the settings, you can add more and more filters to that are suitable for different cases. For example, you can create a filter for de novo variant. You can create a filter that eliminates every variant that is homozygous in a parent. So the filtering capability in Genix is very flexible and aiming to help you analyze as quickly as possible, which is the main target of Genix. So let's just start reviewing this variant list that we see here. Now, we said that this is a case of a child with a suspected syndrome. And in order for Genix to help you analyze the case, we have the phenotyper. I think I've already discussed or elaborated about this. The phenotyper correlates genes and phenotypes. And currently this is empty because we didn't put any phenotype for this case. So if I want to add a phenotype, I would go to the orange bar, and under the phenotypes, I would click on this pen. Now, there are a few options or ways how to insert different phenotypes here. If you are restricting yourself to HPO, you can use this. For example, the first feature was microcephaly. I can write it here, search it, and find the correct HPO terminology or term with the HPO code. And if this is the term that I want to add, I can click on Add Selected, and this would become the first phenotype. There is also another option that we called Advanced. The Advanced is when you have a list of phenotypes somewhere in an Excel file, in another system, whatever. You can just copy paste it as it is, and we would use it for you or add different phenotypes for this patient. The only thing that you have to make sure is that you use this format when you want to separate different phenotypes. Now, Genix is not restricted to HPO. You can actually put any medical keyword, any treatment, any drug, different phenotypes. Now, when you want to separate different phenotypes, this format when you want to separate each drug, and this would work perfectly fine because we understand that HPO although this is the most common medical keywords for geneticists and the one that standardized the phenotypes worldwide, we still want to be able or to allow flexibility for the user to use different terms, and sometimes not all phenotypes recapitulate very well in HPO. So this is the list of phenotypes that I had, and now I can click on Save. And while doing that, while updating the phenotypes of this patient, the lists of variants for this patient would be sorted based on the phenotyper score. Now, this is an automated feature that we have, but you can modify it as many times as you want through the analysis. So let's say you are analyzing a case now with a child with autism, and then a year passed by, and then the patient come back, and now the patient describe or display different phenotypes or additional phenotypes. You just open the analysis. Of course, you can update all the annotation sources, but then when you click on the phenotype, you can just change it, and then the phenotyper score would be updated live on the platform. Now, this is very interesting. As soon as I put the phenotypes, now I got a different list of highlighted variants, I would say. And I can now start to review the genes or the variants in the genes that are found with the highest correlation between the phenotype and the gene. If I click on the phenotyper score, I would get a list of evidences. Now, this list of evidence is what the software, what this feature is built upon. So we use text mining directly from many different sources. This is CleanVar. This includes SlateVar, UniProt, OMIM, and Trez, publications from PubMed, and then many other data sources to try to help you to correlate very fast the gene and the phenotype. From my own experience as a clinician in the lab, the phenotyper is very accurate. If you see a correlation which is high, for sure there is a very high correlation between the phenotypes and the gene, and you can obviously use that as a trusty information while reporting. If it's blank, if there is no clinical score, phenotyper score at all, most chances this is indeed not a correlated gene, or this is a novel gene that was just recently described, or just a novel gene as a disease causing in human. So you always have to bear these in mind, these options. So once I've clicked once I've added the phenotyper, and now I have the phenotypes, and now I have the phenotyper score, I have this list of evidences, list of variants, and I can start reviewing one by one. Of course, up to bottom. And the first variant here is in the gene name SMUD6. As part of the Genix platform, you have many different pop-ups. We separate different screens with different set of information for the ease or the convenient analysis process. So if you click on the location, for example, this would take you to the IGV, so you could see the reads with your own eyes. And this is obviously only when you start with a FASTQ file, you could see your own reads, the reads of the data. And we also have the gene pop-up, gene screen. And if I click on the gene, I would get this visualization, which summarizes all the relevant information on the gene level. So this is not on the variant level, this is on the gene level. The first panel is the constraint parameters from gnomAD and from ClinGen, if you remember, haploinsufficiency score and triple sensitivity. The second region here would contain information from OMIM or phenotype information. And then you have a summary of all the pathogenic and likely pathogenic SNVs in CleanVar for this specific gene. Now, this is very important because sometimes you have a specific effect in your variant, let's say missense variant, and then you want to know whether missense variants previously were reported or classified in CleanVar for this gene. So this is where you can assess this type of information. We also provide you external links. So you have an external link to GeneCard, OMIM, gnomAD, CleanVar, and GTEx if you want to have expression profile for the gene. Every link here would send you directly to the gene page, and it's not a general link. In this upcoming version that we have in 1 week time from now, we also added the OMIM synopsis, the clinical synopsis by OMIM. So you have the list of OMIM phenotypes, and if you click here on the plus, you would get the description, the phenotypic description of this gene based on the literature and based on the publications that were curated by OMIM. And we also have the cipher. This is just the first tab, which is disease and phenotypes. The second tab would be expression profile. Third tab would be pathways. So, for example, this specific gene takes a part of the TGF-beta signaling pathway. We also have coverage information. If you start from a FASTQ file, then we can assess the coverage per exon. And this is very important when you don't have enough coverage for a gene and you want to know which exons are not covered. For example, if you suspect that there is a compound heterozygous in a recessive gene. And there is also a gene description that you can fill in from the settings if you want to have descriptions for the genes. And this will go to the reports and be part of your report. as an additional information. Now, was just one example on the SMUD6 gene, and as you can see, this gene received a very high score of the phenotyper score. The gene is, if we go back for a second, causes a few phenotypes by OMIM in an autosomal dominant manner, and this is a heterozygous variant. Now I can start to review and assess different annotation sources in my table. The first thing that jumps to my eyes when I look at this variant is the fact that under the QNR panel, which is the quality measurement of this variant, it was called as low QNR. The QNR is an overall measurement that we provide in GenX, and this is based on several parameters of QC data. If I expand this, you can see we have the GQ, we have the DP2, which is the coverage of both alternative and reference variants. You have the percentage of alternative to show you the amount or the percentage of the reads that showed the alternative variant. And you have a PL score and AMP score. And all of this information, besides the first column, the QNR, comes directly from the VCF. So this is a variant that was called in only 2 reads out of 8, which is a very low number. This is below the threshold for reporting, obviously. And for that reason, I'm not going to waste my time and try to better assess it. This brings me to the second row here. And this is a homozygous variant. This is actually a hemizygous variant because it's on the X-linked. If this was defined, if you define the gender as male, this would be referred as hemizygous, not homozygous. So what we see here is a hemizygous variant on the X chromosome in a gene named PQBP1. If I review very shortly, I can see that the reference variant is CAGAG and the alternative is C. This means that there is a deletion of the AGAG nucleotides. And obviously, as these are not divided by 3, this is not in frame, this would predict it to cause a frame shift. Now, if I go here under the effect, this is the effect of this variant. It's indeed a frame shift variant. The zygosity of this variant is homozygous. As I said, the QNR is high, which is very important. Almost all reads showed the alternative allele, almost 100%. And the phenotyper score was very high. If I click on the phenotyper score, I would see the matched phenotypes. So that was microcephaly and the dysmorphism, the ASD, delayed milestones, and all of these features were thoroughly displayed in different sources as correlating highly with this gene. And of course, you are welcome. There are links here, so you can jump to the different sources and try to assess it yourself. The next screen that I want to show you is the ClinVar one. And as we discussed ClinVar thoroughly in the last session, if you click on the information from ClinVar, you would see that this variant was classified as pathogenic. You see here clinical significance by different labs multiple times. So this is actually very encouraging in terms of a diagnosis for this patient. I usually jump into the ClinVar page and get to see right over here the VCVs, which are the variant submission information, and I can see the different labs that have reported this variant before. I can see the GeneDx, for example, which is a large lab, and many other submitters are here. Now, as this was classified, uniquely or altogether as pathogenic and not conflicting. Sorry. This is actually quite convincing as a pathogenic variant. And I can see also that it was classified under different conditions. But I do see that this gene causes Renpening syndrome which is a syndrome that fits the clinical presentation of my patient. Now, if I click on this specific gene, as you remember, I would see the OMIM phenotype, and I can also open and read the description of the syndrome and the different parameters that usually comes with the syndrome. Really easily, I could see that ASD, atrial septal defects, are part of this syndrome, and some face, ears, and eyes abnormalities, which will all go under the facial dysmorphism of this child, and also microcephaly. So there are in short stature, which was also mentioned in the referral letter. So we understand why there is a very high correlation between the gene and the phenotypes. So this is the OMIM and the OMIM inheritance. This is an X-linked recessive disorder, and that fits with the fact that it's a homozygous variant in a male patient. The next column that I want to show you is LITVAR. LITVAR provides publications that are on the variant level. It's a great tool by NCBI. If I click on it, I will jump directly to the variant page, and I would get to see the actual publications of this specific variant. This is very, very helpful for providing a reference in your report, for reading more of different patients with this variant, etc. Now, the next panel is the in-house. As I said, this specific account is completely empty from cases, so there is nothing here. But basically, we provide the allele frequency from the in-house database and information based on the variant and on the gene level. So if this specific variant was classified as pathogenic in my own account before, I would see it here. So in the next panel of effect and prediction, if I expand this now, I can try to assess the algorithms or the prediction tools that try to predict the specific variant. Now, this is a frameshift variant. I don't have here a CAD score. This is an indel and not a missense variant or an alternating variant. But I do have information from the impact information, which is high impact. The PLI of this gene is high, close to 1, but not 1. And this means, just to remind you, that this means that this gene is less likely to have a loss of function on a heterozygous state. I have the SpliceAI information or score, which is a bit low. That predicts that most chances this is not a splicing variant, donor or acceptor. And the last category of annotation source would be the frequency. Now, I see here from GNOMED, this is just blank. So there is no any person or individual in GNOMED carrying, and even as a carrier, carrying this variant. So overall, I found a hemizygous variant in an affected child, which was previously classified in ClinVar, and we also saw some publications on this specific variant before. At this point, I'm convinced that I have the diagnosis. It doesn't mean that I'm not going to further review other genetic models or other variants, but let's say I've reached my diagnosis and I want to report it. I would click here on the left panel. I would choose the inheritance model. This for in this case, I can choose between recessive or dominant. Doesn't really matter. Don't really need to specify this if you don't want to. The relevance. So sometimes we report few variant of unknown significance, and we want to grade them with their clinical relevance or with their relevance to the patient. You can do that using this section. And then the pathogenicity. Now, the pathogenicity is not an ACMG classification. This is the in-house, the internal lab classification, which combines information from ACMG and from other type of information like previous knowledge and segregation in the family if you have, etc. We are not going to describe thoroughly the pathogenicity now, as we have dedicated classes for ACMG classification. in this specific case, I can say that this variant for sure is classified as pathogenic. The next section here is validation. I can state as a lab whether this variant requires validations or not, whether it had a positive validation. I think in most labs today, people are not validating exome sequencing data or variants if the variant is sufficiently covered. But sometimes the lab would issue a sentence saying, this variant is there, but there is like low QC or low number of reads, and I recommend you to validate it by another method, which usually is a Sanger sequencing. The next section is notes. So usually every lab would have its internal notes, and this is information that you want to save for downstream analysis. So let's say I'm investigating in this case, I'm reading on the variant, I'm classifying it, and then I have some notes for my own team to know. And that's very important for two reasons. First, when I go back to this case in a year from now, that would help me dramatically to understand what was the exact situation. Usually under notes, you can write, I don't know, the mother is affected, for example, or this requires validation if you want to just write it again. Or there's like low QC, or if you want to just highlight the annotation sources that you used for the pathogenicity. For example, splice AI was low and conservation was low. So such parameters that you want to provide as notes that highlights what was important during your analysis would go here. The notes box here is internal to the lab. This is not going to the report, so you can write whatever you want here. The tags is the next section. We allow users to tag variants. For example, de novo, founder variants, incomplete penetrance, low QC. And this is in order for you to be able to query such variants in the future. So some users ask to pull all the de novo variants. So once you tag a de novo variant, all the de novo variants, this makes it possible. The tags, there are default tags, which you see here, but this is completely flexible. You can go to the settings, add, remove tags as you wish if you have your own lab tags for variants. The next tool, the next box here is a variant interpretation box in which you can write the interpretation of the variant. This would go directly to the report. And there are actually two fields, variant interpretation and variant level recommendations. And you can use these two boxes to just capture some specific information that you want to put on the report. After that, we provide a area in which we specify the different ClinVar submissions. You can select these submissions, and then they would appear in the report. Some labs provide information from ClinVar in the report, some don't. Of course, up to you to decide. And then you have the OMIM section, and if I click on it, all the information that comes from OMIM, including the description of the gene, the clinical synopsis that is relevant, publications that are relevant for the genes, everything would create a section in the report for the OMIM information. And there are some evidences from the literature that you can also add to the report, but this is less recommended. So usually we put OMIM, and in some labs, you also put ClinVar. Now, once I click save, I also have this is high relevant. Let's put a tag here. This is none of the above, actually. This is just a well-defined. Oh, sorry, I didn't click on save. A well-defined previously pathogenic variant. So this is going to be very clear diagnosis for the patient, for the referring physician. I think this was quite clear. This patient has Renpenning syndrome. And at this point, you can, once you click on the relevance, once you select the variant, this would go to this upper link here. If I click on it, this is the selected variant screen. and only selected variants would go there. Usually before I go to the report, I just make sure that the variant is there, all the variants that I wanted to report on. A few other information or just an instructions on this dashboard that we see here. In the upper panel, you have links to the subject page, the analysis page, and then you can also play with the open status, the closed status of this case. So once you've finished, now I'm finished with this one, I can click on closed, and then I can also assign it to specific users in the account. I'm the only user in this account right now. This is not an option. And then there is info on the subject level. You see here, we didn't specify the date of birth or the consanguinity or the ethnicity, but there is an option for you to fill this in. The clinical information would contain all the phenotypes that we used at the time of the reporting. So I can play with it as much as I want, but to the report, the phenotypes that were at the time of the report would appear. And there is some VCF information, QC data that comes from the VCF, how many variants, how many amino acid change, etc. What is the genomic build, etc. And by the way, all the cases in this course would be in AG19. There is also a magnifier here. Using the magnifier, you can search for a specific gene if you just want to see all the variants in this gene. So I just, as an example, put here an FMR1 gene, and I can see the lists of all the variants in this gene, regardless of all the filters. So for example, the first one here has a 100% allele frequency, and it appears here in the table. The magnifier is very helpful when you have, when you know what will be the affected gene, or when someone asks you about this case, saying, sometimes it's like the partner of the sample has a carrier, a positive variant in a specific gene, which is recessive. So she or he are carriers. And do you see any carrier findings in your own sample? And then you could use the magnifier. There are also settings here. So filters in this table, you can sort just by clicking on it. I can sort all the genes descending, and I can really control like an Excel file. So if you see here, there is like a small filter icon. This is almost in all columns, and you can just decide, just like in Excel, which types of values in this column you want to see. So for example, if I go to ClinVar, I can deselect some of the classifications, and then the variants would be removed. So the column filtering and the sorting is something that you can do live on the table, makes it a dynamic process of the analysis. And if you just want to revert when you play with the variants and you want to see the effects and you filter out, filter in variants, if you just want to reset it to the default settings or to the default state when you open the analysis, you can just click on the settings and click on reset analysis filter. This would think for a second and then would bring you back to the original status of the filtering. More options here would be to reset the column width. Sometimes this could change from one screen to another, so you can use this to adapt to your own screen. And you can remove all the column filters. So this is another option if you just want to unfilter specific column filters. Okay, so I think with that, we are getting to the end of this case. And I know there are a lot of things that I haven't explained, but I'll do that in the second case. I think we have...
Thank you so much, Hagar.
Jade, I think there are a few other slides that you've missed. Can you please push start again? Sorry, everyone. Thank you for the questions in the meantime.
Give me a second. I'm going to do that right now.
Okay, no problem. Key features of the syndrome is that it causes developmental delay and moderate to severe intellectual disability. The physical features also include short stature and microcephaly, and the facial characteristics are long, narrow face. So previously, the physician described this child as dysmorphism on a general thing, but obviously there you have to specify, the physician must specify what exactly is the dysmorphism. And in this specific syndrome in child, this includes a narrow face, a bulbous nose, which is this firm of the nose, and other specific dysmorphism that we can see in this child. Also, another characteristics of these patients are small testes in the affected males. The syndrome is inherited as an X-linked recessive, primary affecting males, but females are carriers and rarely could also be affected when there is a skewed X inactivation that eliminates the expression of the normal healthy allele. In terms of the genetic characteristics of the syndrome, this causative variants in PQBP1 gene that is located in the X chromosome are the causing variants for this syndrome. This gene, the PQBP1, function as or codes the polyglutamine binding protein 1. This binds to stretches of glutamine in other proteins. We can see it right here in this pathway scheme. And the role of this protein assumed to participate in RNA processing and transport, affecting gene expression in neurons, and this explained the neurological features of the patients. In terms of the localization of the protein, this protein is found in RNA granules within the nerve cells, aiding in RNA transport and storage. And as we already said, causative variants in this gene are associated with the Rennes-Penning syndrome. So with that, I want to finish this case. I hope you learned and you got some tools to work with when you approach to a new analysis. Before I go to case number 2, I want to stop here for a second and ask or answer some questions.
Thank you, Jade. So we're going to take some questions right now. I'm going to share my screen. Not this one. Okay, so thank you for all the questions in the chat. We're going to try to address them all today, and I hope that we can make it. We have some time. So the first question, well, can you see my screen, right? Hopefully you can. First question was about follow-up questions from the first lecture, and that is, assuming we have 2,000 filtered variants in the annotated VCF, what check could be besides IGV to ensure that we don't have sequence or technical artifacts? And are there any tools that can help us distinguish between an artifact and a real sequence or a real variant? Well, the answer for that is quite a few elements that we can use. First of all, is the QC parameters of the variants. So every variant in the variant calling has a QC parameters that help us to determine whether it's a real call or not. This is very helpful, and there are some guidelines or thresholds that are recommended by the companies as real variants versus unreal variants. And in some of the variants in the filter field on the VCF, you would get a word saying pass. So if you have a pass on a variant, that usually means the variant is probably real. The second way of trying to assess an artifact of a sequencing would be your internal database. So once you start aggregating a lot of samples and you have your internal database of, let's say, about 100 samples, then most of the variants would start to repeat itself. And if you have a variance which found in 90%, for example, of your local database, it's either real and very common, or it's just an artifact, a sequence artifact. So I hope that was helpful. And I think this is like to distinguish between an artifact and a real call is something that you build with time. It requires some experience of the analyst, but once you review several dozens of cases, it gets to be more and more easy. It's more complicated for copy number variants, but this I'm going to leave for Ali's classes down the road of this course. There were a few marketing or business, I would say, questions about minimum license package and price for revision. So if you want to have pricing, we're not going to talk pricing on this course. You're welcome to write Jade an email. We'll get you the price list that is relevant for you. And that, of course, are really dependent on how many samples are you going to analyze. Regarding a revision, so just for you to know, revision is free of charge, and you can just revise the data. And once you analyze or annotate the sample for the first time, then you can revise it again and again. So going back to some questions from on the platform, how can we add gene names into a panel? Now I'm gonna share my screen and show you right how you do it. So in GenX account, you have the settings on the left side. There is a navigation tool, and in the settings there are gene panels. So thank you for that question, because this allows me to show you more features on the platform. Now, to add a new panel, you click on new. You can name it as much as you wish. This could be, let's say, cardio, just inventing it right now. Put a description, and this is just an internal description, and also a category if you have like different categories of gene panels. And then you can put the genetic models that you want to associate with this gene panel and the lists of symbols. Now I'm gonna just write some genes, like this, okay, and I'm gonna write a false one. Once you can copy paste a long list of genes, comma separated, and click on validate, and then the identified genes would be on the upper panel here, and the unidentified genes would be on the bottom part. And if there is a related synonymous of this gene, you could solve it or resolve it with our suggestions. So then you can click on save. You'll have a new gene panel that you can associate from the analysis. We also address you to Panelab for a recommendation. So you probably heard of Panelab. There is UK Panelab in Australia. I heard, this is just based on rumors, we heard that the Australian one is is updated frequently, is better than the UK Biobank, than the UK Panelab. And you can just search for, let's say, autism, and then click on it and get the recommended curated panel by experts in the field. And you can just download it and have all the information that is required. And here is the list of genes for this. Once you download this list of the genes that are related to autism from Panelab, you just upload them to GenX back to in the settings, and this allows you to just have your own new panel. Okay, I'm gonna stop share my screen and go back to the questions. Another question was, can we analyze mental health? Well, this is kind of a clinical questions that I want to address to Ali, which is here. Are we doing any exome genomes for mental health disorders?
Hi. Sorry. So what I understand from your questions, you would like to ask, is there any specific gene panels we have on the GenX platform for the mental health?
I think that was a broader question unrelated to gene panels, just whether are people doing NGS analysis for mental health disorders.
You mean the question means like they have a specific enrichment kits for the intellectual disability panels, for example, the Parkinson's or the dementia panels?
For example, yeah, go ahead.
So there are plenty of protocols commercially available, and we have on the GenX platform, we have implemented those protocols for the mental health, for the intellectual disability, dementia, Parkinson's, Alzheimer's. All those protocols, they are pre-made into the Genix platform. You just select the right panels from the mental health status, and you can start analyzing upload your VCF files and select the panels and start analyzing your case. It's quite easy in the Genix platform.
Thanks, Ali. I would just add to that if the question was regarding mental health that are polygenic, like schizophrenia, dementia, and conditions that we usually cannot perform NGS, or this is not very, very valuable, then I guess, well, this is where I took this question to. We are quite limited with the knowledge that we have scientifically on mental health disorders of adult onset. So if we're talking about like Parkinson, it's quite easier because we have well-established genes that we know that are correlated with Parkinson. But if we take it to schizophrenia, for example, it's going to be much harder to find a variant that is causing schizophrenia, because usually this would be polygenic, not monogenic. But basically, there is enough scientific knowledge about this. If you have your own panel for schizophrenia, you just deploy it into Genix and you can assess the samples. Another question was, how do we evaluate mutation on pseudogenes? This is a very important question. And do we always confirm it by Sanger sequencing? So first of all, in the Genix platform, we mark genes with a P if they have a pseudogene. So you would get like a red flag saying this gene has a pseudogene, and you better try to assess it with your own eyes in IGV. So this is how it is recommended to do the analysis, so far to do the analysis. Regarding the validation by Sanger, some labs would always recommend to Sanger sequence this validation process for genes with pseudogenes. Some would not. I think it really depends on the specific variant. So there are variants that we know that could be found in the pseudogene or in the real gene, but sometimes just only part of the exons overlap between the pseudogene and the gene. So you can have like assessment trying to say, is this region, micro region in the gene has a resemble sequence on the pseudogene? If you look by IGV, the regions that have multiple or duplicate alignment would be white. So if you see white reads on the IGV, that means that the read was mapped to 2 different locations in the human genome. Such variants, for sure, require Sanger validation. Another question was about compound heterogeneity, and I think this question was about compound heterozygous. So we have in Genix a dedicated tab to show compound heterozygous. If this is a trio analysis, we take only pairs of variants that in the same gene that comes one from the mother, one from the father in the proband. If it's just a single exome and you don't see the parents that are associated, we take pairs of variants, assuming these could be introns or cis, without this knowledge. We also support phasing in Genix. So if you run long-read sequencing, you have phasing information that allows you to distinguish between the 2 alleles. It's also an option to calculate automatically the compound het. And as we have a dedicated tab for compound het, all this information would be calculated and put there. We also have a feature that compares copy number variations to single nucleotide variations. So even if you have a compound het between an SNV and a CNV, this is still something that you can try to assess with our software, as we calculate it automatically for every CNV and for every SNV. Another question was, can we analyze only a single gene data? Of course. So basically, whatever you can sequence, we can analyze. If you are sequencing a panel, not the entire exome data, you can obviously assess or look only in the single gene. You can also have a gene panel that is built of a single gene. And you can also have a virtual panel, meaning you can sequence the entire human genome or just the exome sequencing and then only analyze a single gene and not seeing the entire exome in Genix. So the next question was, can I evaluate multiple individuals more than a family trio, siblings, uncles, etc.? So yes, definitely. Let's say, okay, I'm gonna show how to do it, but this would require a new share from my side. Okay. So on the way. When you analyze or when you upload a single sample to GenX, you can add associated samples. Once you click on add, and I hope you see the platform, once you click on add, you can define the relation of the sample to your program, to your sample. This could be a match meaning it's the same individual exactly. And sometimes when we go to cancer, for example, we want to compare the germline versus the somatic pipeline. We call it tumor versus normal. In such cases, this would be a matched because it's coming from the same individual. It can also have a mother and a father, which obviously is a regular trio, but then there are siblings. It could be a twin, it could be a mother relative or a father relative, or just other, because sometimes we just want to have another control. So these are the currently associated samples that you can specify. And you can associate dozens of samples to a single in a single analysis. So if you even if you have a cohort of samples displaying the same phenotype, you can just put them all in one analysis and try to assess it all together. Okay, so going back to the questions, Ali, feel free to jump in with new questions from the chat because I'm not monitoring this right now. Can I evaluate, so this we went through, is there a way to evaluate degenic inheritance? Well, this is a very, very nice question. We know that there are several dozens of genes that were characterized to have a mode of inheritance, which is a bit different than a classic monogenic inheritance model. And this is the degenic inheritance. And the meaning for this means that there are mutations or variations in two genes, and you need to have the two genes affected in order to have the phenotype. Currently, we do not have an automated analysis to detect these events, but we do display under the OMIN inheritance model, DG for degenic. And if you see a pathogenic variant in a DG gene, you should try to assess if there is another pathogenic variant or likely pathogenic variant in the other gene that comes with it in a pair. I just want to say that in the next few versions, we are planning to have an automated degenic inheritance. This is still quite, I would say, a rare situation in which you find a diagnosis that is degenic. Is only AG19 available, or data can be annotated in AG38? We support both genomic builds completely. And you can upload, when you upload a FASTQ or a PCF file, you just need to specify the genomic build of either. And lastly, before I open again the chat, can we add a panel to the ongoing analysis? Definitely, you can specify the panel in advance. You can associate it to the protocol to have a streamlined protocol with a single panel. And you can just open a random analysis without any filters and then associate a single panel, two panels, etc. So this is not restricted at all. You can associate and deassociate a gene panel at any given time through the analysis. You're welcome. So I guess we plan to have two cases this session, but we're kind of running out of time, and we see the value of answering your questions. So, are there any other questions in the chat?
There is one more question. Can we add more panels in the ongoing analysis?
So yeah, the answer is yes. You're welcome. Everyone are welcome. Thank you for the comments on the chat. You can associate more than one gene panel in one analysis. Sometimes this is beneficial when you want to run for a single sample a few different panels. Like, let's say this is an autism patient with hearing loss, and then these are two different panels that you want to associate in the same analysis. We also offer that in cancer. So in my own lab, when we run cancer panels, we also take thrombophilia genes because there is a medical benefit of knowing bleeding disorders and such conditions which are hematology in cancer patients. And we also do pharmacogenomics that is related to cancer. So in Genix, which is built like building blocks, as long as you know to define what you want to assess, it's very easy to create your own protocol, associate with the gene panels that you want to analyze, and you can also analyze or associate a variant map and not only gene panels. So this, for example, if you have like a list of variants that you want to analyze in every case, these could be founder mutations, these could be just common mutations that you know are very important for such analysis. Then you can associate a variant map and validate that these genes are actually there, reference, etc. I guess I don't see any other questions, which is good. That means that we gave enough time today for questions. Anything else, Ali, Jade? Ali, you're muted.
I think.
I see
Yeah,
see that there's one more question here. I don't know, did you answer? Can you save the different panels or gene lists?
Yeah, of course. So you just save them in your account and you can save endless number of gene panels. You can have them versioned. So if you have if you offer in your lab one panel and then you put version 1, then you can add versions as you update this gene panel. Also, when you associate a gene panel in Genix, all the genes that are found in this panel would go automatically to the report under a section of gene panels. So this is just part of the reporting system in which you know exactly which genes you reported in this specific analysis. So yes, definitely these are saved.
Great. Thank you so much. I think everybody's getting a lot of knowledge and information and, really excellent knowledge in this course. I see so many